Hello everybody, my name is Elliott Kimmel and I'm a high school teacher at Central Secondary School in London, Ontario, and the course we'll be talking about is grade 12 biology, the photosynthesis unit. Now we're close to the end of the photosynthesis unit and I want to do something called photosynthesis odds and ends, and I will be talking about the C4 cycle, the CAM cycle, and comparing photosynthesis and cellular respiration. Now, the textbook reference is Nelson Biology 12, and so if you have that textbook available, I'll be showing you the page numbers and you can follow along, or if you have diagrams or worksheets, you can use those. So here we go. This comes from Nelson Biology 12, page 169, and I'd like to talk about the C4 cycle. Now, we've covered the light-dependent reactions and the light-independent reactions in the C3 cycle for the latter, the Calvin cycle. And the C4 cycle is a modification of the C3 cycle, or the Calvin cycle, and this is an adaptation to hot, dry climates. So when you consider the weather being hot and dry, plants need to close their stomates so that they don't lose too much water, right? We've got a stoma down here, and whenever the stoma is open, water tends to exit the plant, and carbon dioxide cannot enter the plant, and so this is a problem for photosynthesis, right? So in the hot, dry climates, in order to conserve the water, which is a main consideration, the plants are going to have to close their stomates. So how are they going to deal with the fact that, first of all, very little carbon dioxide is coming in? I mean, the stomates do open, but they open infrequently, right? So how are they going to deal with not very much carbon dioxide coming in, and also the fact that oxygen is being produced by the light reactions, and the oxygen is building up inside, leading to something that I mentioned over here, photorespiration. So we have to deal with that. So C4 cycle, or C4 plants that have the C4 cycle, is an adaptation to this kind of condition. And this is the kind of thing you're going to see in the tropical plants and plants that are grown out on the prairies and stuff, when it's very hot in the summertime. These are the plants that manage to thrive in those environments, whereas a C3 plant, you know, a maple tree or an oak tree or something, is just not going to do very well. All right, so we have some physical or structural adaptations, and we've got some physiological or biochemical, metabolic adaptations to deal with. So here we see an image of the C4 leaf anatomy, and the main thing that is different in these C4 plants is that there are bundle sheath cells here all surrounding the vascular bundle, the vein, all right? And we also have mesophyll cells. So typically the C3 cycle was happening out in the mesophyll cells of typical C3 plants. But in the C4 plant, we have this arrangement where we have compartmentalization of the metabolic processes, with something going on in the mesophyll and something going on in the bundle sheath. All right, so we've got two sort of cycles happening together. And really, when I talk about the C4 cycle, which is illustrated here, I'm actually referring to the fact that there are two cycles. So there's a C4 cycle there, and there's a C3 cycle down there. All right, so here's what happens. Stomates are closing and only opening infrequently. In order to survive, a plant is going to have to be able to grab whatever CO2 is available, whatever minimal amount of CO2 is available. It's going to really have to be efficient in grabbing that. The problem with the C3 plant and the enzyme down here called rubisco, the problem with that is rubisco can grab either CO2 or oxygen. And if it grabs the oxygen, if there's little CO2, there's not much of this, and there's lots of oxygen, high O2, we get this process called photorespiration, and this is bad. So what's going to happen is when there's little CO2, we need an enzyme, and the enzyme is down here, PEP carboxylase, with very, very high affinity for CO2 and low affinity for oxygen. And this is what happens in a C4 plant and in the C4 cycle. The limited amount of CO2 is efficiently grabbed or fixed by PEP carboxylase onto PEP. Now, PEP is phosphoenolpyruvate. It is a 3-carbon molecule, so with the other carbon from carbon dioxide, you get 4 carbons, And that is the first organic molecule produced when carbon dioxide and PEP unite through the action of PEP carboxylase. A 4-carbon organic acid called oxaloacetate, OAA, is produced. So that's why this is called the C4 cycle, because the first molecule that includes the CO2 is got 4 carbons, oxaloacetate. In the C3 cycle, the first molecule that includes CO2 when it clicks onto RUBP by the enzyme Rubisco is a 3-carbon molecule, so it's the C3 cycle. All right, so you're closing your stomates because you're a leaf, assume you're a leaf, and it's very hot, and you need to conserve your water, so you close your stomates, and there's limited CO2. So what do? You use a very strong enzyme with high affinity for CO2 to grab whatever little bits of CO2 there are, click them onto PEP, fix them onto PEP, and you make OAA. Now this happens out here in the mesophyll cells, all right? Oxaloacetate then is converted into another 4-carbon organic acid, which is called malate or malic acid. Now, in class, we talked about this process, oxaloacetate, and we said the oxaloacetate then moves into the bundle sheath cells. And that came from a university textbook that omitted the malate. Well, in this textbook, we have the malate. In the Nelson textbook, it talks about the malate, so let's go with that. Oxaloacetate is converted into malate, and malate then moves from the mesophyll into the bundle sheath cells. Now, these bundle sheath cells are special in that they do not allow a high concentration of oxygen to exist in there. So we can safely have the wimpy enzyme Rubisco, which is a very famous and very prominent enzyme, but it tends to recognize oxygen and fix oxygen instead of CO2. If CO2 is low and oxygen is high, it grabs the oxygen and photorespiration occurs. Well, in a C4 plant with this particular type of anatomy, the Rubisco can exist deep down in the leaf, in the bundle sheath cells, where oxygen is prevented from accumulating, so that malate can come down here, go into the bundle sheath, and release the CO2, and then it now exists as pyruvate. All right, we've got 3 carbons here, 1 carbon there. There's the 4 carbons of malate. Pyruvate then will go back up into the mesophyll and be converted back into PEP so that it can join with more CO2 through the action of the enzyme PEP carboxylase. This process allows carbon dioxide, whatever limited amount there is, to be shuttled into the bundle sheath cell where it starts to build up. And we get a little bit, and then we grab a little bit more. And as the CO2 builds up, that's my CO2 building up, as it builds up, this enzyme Rubisco is now able to grab just the CO2 and not oxygen, and we can go through the typical C3 cycle or Calvin cycle and produce our sugar. All right, so that is how the C4 cycle works in tandem with the C3 cycle, but these are called C4 plants because they start with the C4 cycle and then we have the C3 cycle. This is an adaptation to those hot, dry climates where the stomates close, and the idea is to avoid photorespiration. This is what is necessary. In the more temperate, wetter climates, a C3 plant is just fine because the stomates can open frequently. You can get rid of your oxygen. You don't really worry about a lot of transpiration of water. I mean, some will happen, but not too much, because it's moist and it's cool, and you can get lots of CO2, and therefore the C3 cycle works just fine, and they don't even have the C4 cycle, all right? But in the hot, dry climates, they do, all right? So there's a little review of the C4 cycle from page 169. All right, we then have the C3 cycle, the C4 cycle, or C4 plants, C3 plants, C4 plants, all right? And here you can see in this diagram from page 170, 171, we've got C3 plants that exist fine in cool, moist climates, and we can see how CO2 or oxygen can bind to Rubisco, All right. And of course, if photorespiration occurs when it's hot, then this is what's going to happen. This is a bad process, this photorespiration. The adaptation then, therefore, is the C4 plants that exist well in hot, dry climates. In a cool, moist climate, this is okay, all right? We're not going to get a lot of photorespiration. This is saying what happens if you took a C3 plant and you had it in a hot environment, then this would happen, and that's a bad thing. But in a hot, dry environment, C4 plants are going to work out fine. The CO2 is going to be converted to oxaloacetate. You know, and again, see the way they don't even really include the malate. The malate's just an intermediate, all right? And then they show how the Calvin cycle will happen, and they show how there's a separation. You got the mesophyll cells here, you got the bundle sheath cells, and reduced photorespiration. So that's how that would work. Now, there is another type of photosynthetic process, which is called CAM, crassulacean acid metabolism. The A stands for acid because organic acids are going to be made just like in this process, the C4 cycle. But this is what's happening in stuff like pineapples and cactuses and stuff, you know, desert kind of plants. If it's very hot during the day, a plant is going to have a lot of trouble opening its stomates at all right? And even the C4 cycle is not going to go well. You're not going to see a lot of like palm trees or banana trees in the desert, all right? They're just not going to survive. You'll see them in the desert oasis, or if you're seeing a mirage or something, all right? But if you really want a desert plant or something along that line, then the CAM system is what works for them. And in the CAM system, what happens, let's go down here first of all right? CAM plants, they exist in hot, dry, and desert environments. So, of course, there's going to be some overlap here. But if you're really looking at these harsh environments, what's going to happen is the stomata will open at night, and that's when the carbon dioxide will come in. There's going to be little water loss at night because it's cooler, all right? And the CO2 will come in at night, not during the day. And that carbon dioxide, let's go back up here now. The carbon dioxide, similar to the C4 cycle, will be converted into an organic acid, all right, like oxaloacetate or like malate, whatever. But in the daytime, this is when the stomata will close to avoid transpiration. And that's when the carbon dioxide will be released from the organic acid in the daytime, stomata close, CO2 is released, and now the dark reactions, the Calvin cycle is going to occur because, of course, in the daytime, that's when you have the light reactions will occur. You'll make your ATP and NADPH. You don't have that at night. So, you know, if you were a desert plant here, and you open your stomates at night because it's cooler and you get your CO2, you don't actually have the ATP and the NADPH to do the whole process of photosynthesis. So take your CO2 in at night, store it up as an organic acid, and then release the CO2 in the daytime. Close your stomates, you already got your CO2, make some ATP and NADPH, and now you can use that CO2 in the Calvin cycle. So that's the modification for these CAM desert plants. Get your CO2 at night, close your stomates in the day, all right? And then fix your CO2 during the day in the Calvin cycle. All right, let's move on. Now, this is Nelson Biology 12 on page 181. I want to look at comparing photosynthesis and cellular respiration. Those of you that are in my classes at Central will have this worksheet, and hopefully you'll have this worksheet completed. What we need to do here is compare photosynthesis and cellular respiration, look at some of the things that are similar and things that are different between those two metabolic processes. So table 1 does a comparison of the overall reactions. So let's look at the reactants now. In cellular respiration, the reactants were organic molecules, and in particular glucose, which will go through that central pathway of glycolysis, and then of course we'll have matrix reactions and the ETS. All right, so the major reactants, and we know that ATP is necessary to provide energy, and we know we need NAD+, and all of these things could be considered reactants, but the major one is glucose. Whereas in photosynthesis, the major reactants, of course, are carbon dioxide and water, and the carbon dioxide is going to be used in the dark reactions, the light independent reactions, the real source of carbon to produce the organic molecule that we're going to make here. And the water, of course, is primarily used in the light reactions, photolysis, source of hydrogen ions for the proton gradient, oxygen, well, that's just a byproduct, but electrons to regenerate the chlorophyll A's in the photosystems. The products of cellular respiration, as you know, are carbon dioxide and water. The carbon dioxide primarily released in the matrix reactions, in that preparatory step or conversions, and in Krebs cycle. And then the water being produced at the very end when electrons are transferred from cytochrome oxidase onto oxygen, the fine electron acceptor, producing water. So major product. Yes, of course, ATP is produced, but we're really looking at these molecules here. In photosynthesis, organic molecules such as glucose, the reverse here is being produced. When you look at the bottom of the C3 cycle, the Calvin cycle, you'll see the glucose there. Of course, you may see starch as a product, or cellulose, or sucrose, but all of those really are either polymers of glucose or isomers of glucose, or some modification of the glucose. In terms of energy for these reactions, energy is released by cellular respiration by the breakdown or oxidation of glucose. Energy is released, so this is an exergonic reaction, whereas photosynthesis, energy is stored. You take the energy from sunlight coming in and you kick electrons around and you make ATP, and you use that ATP and some NADPH to fix the CO2. So really, energy has been stored in the final product of glucose, the energy that originally came from the sun. Exergonic, endergonic. Okay, let's talk about electrons now. The source of electrons in cellular respiration comes from the organic molecule, from glucose. You know that as glucose goes through glycolysis, there are points at which electron carriers come along and oxidize the glucose or the intermediates in the pathway, producing the reduced electron carriers here. So they are coming away carrying those electrons. So that's the source of electrons. In photosynthesis, the source of electrons is water. In the photolysis reaction, those electrons regenerating the pigments that have lost electrons. So the electron carriers, we need something to carry those electrons, are NAD plus and FAD. Those are the oxidized forms, and the reduced form would be NADH, FADH2. The oxidized electron carrier in photosynthesis is NADP plus. This is produced in the light reactions and will go over to the Calvin cycle and reduce molecules as the CO2 comes in, and we go around the cycle, we churn off our carbohydrate, and we come back, and we need to do some reductions there, add some electrons and hydrogen, that kind of thing. All right? Okay, let's look at the electron transport system itself. The energy profile describes how the energy is changing over time in cellular respiration versus photosynthesis. You know that NADH or FADH2 drops off electrons to an ETS carrier. All right, let's say it's NADH dehydrogenase. And as the electron travels from component to component in a series of redox reactions, it releases energy. That energy is used to pump hydrogen ions from the matrix to the intermembrane space. All right, so energy is constantly being released from those electrons, and when the electrons get to the very end, cytochrome oxidase, that's when they're going to jump onto the oxygen, and they've given up all of their energy. So it's a relatively linear process of releasing energy. Now, in photosynthesis, we looked at this diagram, and it's called the Z scheme, although I don't have that. It looks more like an M on the side, doesn't it? Anyways, all right. We looked at this diagram and we saw that there would be a light harvesting complex there, a photosystem 2, and a light harvesting complex there, a photosystem 1. And we saw a photon would come in, hit the antenna pigment, etc., etc. Energy would be transferred until it hit the chlorophyll A there, and electrons were boosted to a higher energy level, but they didn't drop back down. They went to an electron acceptor. The electron actually had increased in energy as it goes from the chlorophyll A to the electron acceptor. The electron then went down an ETS, releasing energy, right? And that energy was used to pump hydrogen ions from the stroma into the thylakoid lumen, all right? So opposite that in cellular respiration. And so there was the release of energy, and that's what that energy was used for. Those electrons then regenerated the ones in photosystem 1, and then another photon coming in. This is getting to be a messy diagram, right? And the electron would go up, okay, so it gained energy, and then it would go down in ETS again, giving off its energy. So we have a different energy profile. The electron source means where the electrons are coming from now. Originally, the electrons are coming from here. It almost seems like this is, a contradiction. We got source, and this one says glucose, and this one says this. All we're saying is, in order to create the energy to travel down the ETS, the electrons are being provided by the reduced electron carriers. And of course, in photosynthesis, the electrons are coming from water. Electron sink means where the electrons end up or where they go to. So the difference between source and sink is a common thing that we talk about in sort of chemistry and biology. The final electron acceptor for the cellular respiration process is oxygen, as you know. The electrons go on with some hydrogen and we produce water. So oxygen is the sink or final electron acceptor. Now, I find this a little bit inconsistent, and maybe you will. Let's see if you agree with me, everybody. All right. Final electron acceptor, oxygen over here. Final electron acceptor is not NADPH, it's NADP+. Now, I don't know why this textbook has done it this way. Tell me if do you find this confusing? The final electron acceptor is oxygen here. The final electron acceptor or sink is NADPH. I think they should have said NADP+. I think that would have made it easier for you to understand and easier for me to explain. In the end, the electrons go into oxygen. Well, they don't go into oxygen, but something like that, right? And the electrons are found in NADPH. Really, maybe what they should have done here, just to be nitpicking, they should have put water here, okay? The electrons will end up in water. The electrons will end up in NADPH. Anyhow, I digress. The products of cellular respiration, ATP, of course. The products of photosynthesis, the light reactions, ATP and NADPH. And of course, these are the products made by the electron transport system. So there may be other molecules. Do you want to look at the products here, CO2, H2O. We're not talking about those, obviously. All right, that's an overall. But with respect to the ETS, the product that we care about is ATP. Now, NAD+, there you go. That's also produced as the electron carrier drops off electrons, but that's not considered an important product in this case. All right, moving on. Table 4, ATP synthesis. Hydrogen ions are pumped by the ETC or ETS, electron transport chain, electron transport system. There is one of these or multiples of these in cellular respiration. So yes, hydrogen ions are pumped. Hydrogen ions are pumped here as well. ATP synthesis by chemiosmosis. All right, here is that weird term that talks about the movement of hydrogen ions from a high concentration moving down the concentration gradient to a lower concentration. All right, so this is the movement of hydrogen from the intermembrane space of the mitochondria back into the matrix. All right, that's called chemiosmosis. It's traveling through the ATPase complex. That happens in cellular respiration, and it happens in photosynthesis when the hydrogen ions build up in the thylakoid space or lumen, and they move back into the stroma through that ATPase, and that's when you make ATP. That's ATP right there. Very weird ATP. All right, so it happens in both cases. Finally, organelle structure and function, comparing the mitochondrion for cell respiration and the chloroplast for photosynthesis. And we're focusing on the membrane system. In this case, it's the inner membrane, and that's not the inner membrane of the chloroplast, but it's the very innermost membranes, isn't it? All right. Inner membrane functions. There is electron transport in both cases, as we know, along the cristae or along the thylakoid, along the ETS. There is hydrogen ion transport in both cases, and both of them produce ATP. And these are associated with movement down the inner membrane. Now, this next one contains DNA, ribosomes, etc. for replication. What we're talking about here is replication of more mitochondria or more chloroplasts. Now, we're going to study genetics later on in this year, and you've, of course, already done it in grade 11 biology. DNA, being the genetic material, is not just located in the nucleus, although that is the major repository, how do you like that word, for the DNA, for the chromosomes, but there is DNA also present in the mitochondrion and in the chloroplast. This is called mitochondrial DNA or chloroplast DNA. And this has instructions on how to build more mitochondria or chloroplasts. And the DNA, like I said, are instructions or a code. And just like any code, a code has to be read or decoded or translated. And that's the function of ribosomes. You probably recall that ribosomes produce proteins. And ribosomes, right, they're made in the nucleolus, they travel out through the nuclear pores, they go into the cytoplasm, they attach to the ER, or they're floating freely, and they produce proteins. And the way this works is by a process called protein synthesis. It starts with DNA, which has the instructions or code. The instructions or code is sent out to the ribosome, the factory, and the ribosome makes the protein that the DNA is coding for. All right? And so, look, we're going to get into this process later. This is not really part of photosynthesis, but just to say that both mitochondria and chloroplasts do have some DNA, ribosomes, etc. All right. Location of the hydrogen ion reservoir, the intermembrane space in the mitochondrion, as you know, or the thylakoid lumen in the chloroplast. Notice the opposites there, all right? So there are similarities and there are differences between these organelles and these processes. And of course, ATP is produced in the matrix of the mitochondrion, but in the stroma of the chloroplast. So I hope that helps you understand all of those odds and ends, and I'd be happy to answer any questions if you would like to. All right, take care. See you later.